Welcome to Mobile Lectures. Today we will be talking about the control of duplication in S phase, role of CDK and CDK. So, in case of control of duplication, we have to know that in the G1 phase, there is something forming. There is known as the pre-replicative complex. Now, how this pre-replicative complex forms? Let's focus on that. So we have something known as the ORC. It's also known as the origin recognition complex. This ORC is a key player for the recognition of the I am not that wise, you guys are wise. What? Origin recognition complex, so it identifies origin naturally, but it cannot do that alone. That's the problem. It needs to be associated with some proteins known as CDC6 or CDT1. When these proteins combine with ORC, it forms the ORC CDC6 or CDT1 complex, which along with the MCM protein, I don't know why I cut the M like that, but mistake. So this MCM protein, that's MCM227, so it's a hexameric protein. That hexameric protein actually is a helicase. So this along with ORC forms a pre-replicative complex, which will now help in the replication. Okay. Now this happens in the G1 phase. Okay, the early G1 phase I mean. Now in the late G1 phase, we have, the writing has just disappeared over there. That should be SCDK. This should be SCDK. Now that's SCDK. Actually, while I was setting up the whole thing, I might have rubbed it myself because of my hands. Okay, so anyhow, so that's SCDK. It helps in the formation of pre-initiation complex, and this pre-initiation complex actually helps in the unwinding of DNA, thus DNA replication. Also in late G1 phase, along with the formation of pre-initiation complex, it also helps in something more. That's the disassembly of the pre-RC complex. Now first we are talking about the formation of the pre-RC complex, and now it is conducting disassembly of the pre-RC complex. Why? The answer lies in the cell division. Whenever cells divide, they go through replication, and that replication has to be in very much controlled order. So if there is more than one activation of replication in each cell cycle, that would lead to accumulation of more genes, which may be deleterious to the organism actually. That's why we need to control this pre-RC complex. Thus disassembly occurs in the late G1 phase, or also we can say G1 to S transition phase. Okay, so in late G1 phase, what happens is that there is pre-initiation happening. Pre-initiation is happening also, but at the same time there is disassembly happening of the pre-RC complex at the origin. So at the origin, this pre-RC complex is actually disassembled by destruction of CDC6 or CDT1, thus ensuring only one activation, which ensures only one activation. If more than one activation happens, that is deleterious, I have already told. So this MCM227, okay, this MCM227 is very much important in this step. It's also known as a mini chromosome maintenance protein, not the millimecia protein, but the milli chromosome maintenance protein. I don't play mobile games, my phone is shit. Okay, so this MCM227 helps in the formation of pre-replicative complex, and this pre-replicative complex is like the driver's license. Whenever you are going to drive a car, you need some kind of license. Otherwise, the constable picks you up and thrashes you in the jail and kicks the hell out of you. But in case of pre-replicative complex or the license, he just takes some fine from you. Kala paisa. Okay, we are moving out of the topic. This pre-replicative complex, what it does is that it helps in replication, and also this pre-replicative complex serves as the license for the cell to perform the replication and to move on with DNA synthesis throughout the S phase. Okay, that's what happens, and that is what is the intention of this pre-reparative complex over here. But in this case, obviously, as I have told you, that MCM227, actually MCM protein is very much important. And also remember, in case of exams, the helicase, it's a helicase, and also it's composed of MCM227, that is a 6 subunits arranged in a helical fashion, almost ring-like fashion. Okay, actually every helicase is kind of a ring-like only. Okay, if you study the protein structures, every helicase has a ring-like structure only, ring-like structures. Now we come to the role of MCDK. This should be very brief for this video, as because I have got less time for editing this video, and also I am not editing it. But actually the thing is, I am this takes quite a lot of MB for me to upload this, and I have to make the videos really short. So I will start with the role of MCDK, and this is very brief. Later part I will actually upload in the next video. Okay, the later part of the MCDK. Now this MCDK is commonly known as the mitosis promoting factor, or some other scientists, they like to say it as maturation promotion factor or maturation promoting factors, which you guys are not matured. Sorry. So this maturation promoting factor is synthesized during the G2 phase. What? You go to PP? You are not? Oh, okay. That's my mistake. So they synthesize during S and G2 phase, not when you go to PP. You actually generate urine in your sacs when you go to PP, and okay, what the hell? Okay, S and G2 phase. Now what it does is that it induces chromosome condensation to happen. Now this chromosome condensation, what it helps is that for segregation. That is, suppose a cell contains many okay, let's leave that example. Suppose you are a girl and you have got lots of hair in your head, I guess. I'm very bad in English, so please forgive me. So you have got many hairs, okay? So if you are leaving your hairs throughout the day, like open, you know what will happen to you. Something like fuzz will happen. It will get broken, it will get destroyed, it will get knot-like structures. I don't know what happens to girl, really, I don't know, but actually that is not good for hair. So in this case also, if you are wanting to separate the chromosomes, separate the DNA, okay, you need to condense them first. After condensation, they can be separated. Okay, it's like your headphone. If you are keeping your headphone in your bag, it will get tangled, and then it's a hell lot of pain for you to actually separate it. But if you are actually condensing the headphone, that is, you are tying it around something, or you are tying it around or making some kind of structures with the headphone, such that whenever you take it out, very easily, just by one pull, you can separate. You can actually not separate, then the headphone will be gone, but you can actually open it and listen to your whatever favorite song you want to listen. So it helps in the breakdown of the nuclear envelope. So it also helps in the breakdown of the nuclear envelope. Assembly of the mitotic spindle apparatus. Also it helps in the assembly of the mitotic spindle apparatus, which I am going to cover in the next video. And also for the alignment of condensed chromosome at the metaphase plate for segregation. This metaphase plate is where the chromosomes are lined up in the center of the cells, like you see in many cartoons in YouTube, and which helps in actual separation, which is then separated. Okay, there are some motor proteins. Okay, those motor proteins are actually separating these chromosomes. Okay, these condensed chromosomes. Now MCDKs also serve as phosphorylations. They also phosphorylate wide range of proteins. Those proteins are we are just taking two examples. One is condensin, one is lamin. There are many other proteins which this MCDK actually phosphorylate for the completion of the M phase. Okay, and also for regulating the M phase. The M phase is very properly regulated, I mind you. Okay, we are going to cover this in the next video although. So the condensin. Now condensin is an essential for extensive condensation of the mitotic chromosomes, and I've already told you why the condensation is very much required. And lamin is a stabilizing network. So this lamin protein is actually a stabilizing network under the nuclear membrane that disassembles during mitosis. So this condensin and lamin helps in this. So it's actually the stabilizing network under the nuclear membrane, which during the mitosis breaks, and the breaking is very much required for the nuclear materials, that is the genomic materials, to be transferred to both the cells very accurately. Okay, so condensin is a highly conserved 5 subunit protein complex. We have to know this, that condensin, this upper protein, is actually a 5 subunit protein. Among those, some protein subunits are SMC2 and SMC4. So this condensin is actually made of many SMC proteins. Okay, so there are 5 proteins actually, out of which 2, SMC2 and SMC4, plays a very important role. Okay, so there are many proteins, but out of which SMC2 and 4. I have to make some good arrangements for my videos. Okay. Okay, so SMC2 and SMC4, which are actually playing a fundamental role in the functional organization of the chromosomes. Okay, they actually helps to organize the chromosomes, this SMC2 and SMC4. It is a key football player of the mitotic chromosome condensation. Now, during mitosis, condensin is phosphorylated by cyclin CDK. So during the mitosis, the condensin is actually phosphorylated by the cyclin CDK. Okay, polo and aurora B kinase. So those cyclin CDK, which we are talking about, the MCDK, those are named actually the polo and the aurora B kinase. Polo like actually is also called PLK, polo like kinase. Okay, and aurora B kinase. This phosphorylation of the condensin actually changes the localization and dynamics along with the activity of the protein, that is supercoiling activity of the protein. So actually this condensin helps in supercoil formation. Okay, and also the localization of the nuclear material, that is the disassembly of the lamina, actually the disassembly of the nuclear envelope actually helps it such that this genetic materials comes out of the cell and into the cytosol from the nuclear membrane cytosol, and that helps in actually cell cycle, and also in case of cell division, because of cell cycle we know the cell divides, so in case of cell division it's very much required. Now it also influences the mitotic spindles. Now we have already told you that these proteins actually, that the MCDK, this M-phase MCDK protein, these are not only related to the condensin and lamin, but also they are associated with the formation of many other structures along with many other regulations in the other parts of the mitotic spindle apparatus. Also in some checkpoints, that is a cell cycle checkpoints, you can say DNA repair checkpoints. Okay, also we have got many other mitotic spindle attachment checkpoints or spindle assembly checkpoints. Many other checkpoints are there. Actually three main checkpoints are there, which we are going to talk about in our next video. We are going to talk about this MCDK along with something known as securin separase. So I will get to know what it is. Along with this, I have not written it over there, but we will also talk about the mitotic spindle. Okay, written it. This mitotic spindle I have written. We are going to talk about it in the next video. Thanks for watching. Please like, comment, share and subscribe. Also do not forget to ask any doubt if you have. Also, I may be wrong because I am a human being. So if I have made any wrong during the explanation, please bring it to my notice so that I and you both can learn. Thanks a lot.